Can you see the cursor now? Or did I lose it?
No.
No, you can't see it?
Yeah, we can't see.
Oh, okay. Here we go again. That's my fault for not getting the recording started right away. Okay, are we back?
No.
We're not back. Oh, God. How did I do that? Okay, I'll get us there. Are we back now?
Yeah, we're back.
Okay. Okay, so here we are starting with the digestive system, which we've already covered in theory, and we even had tests on it. But now you're going to be asked to identify certain structures and not forget their functions as well. Okay. The first part of the text here refers to the salivary glands. The salivary glands, there's three sets. The parotid, which are the largest, they're located here on the side of the face. Now this model cuts off the top, but I have other pictures that don't. This is the parotid salivary gland. This is the submandibular salivary gland down here. And the sublinguals are underneath the tongue and do not show here. Salivary glands secrete saliva, and that's primarily used to lubricate food. You've heard this before, but it really will help to let it sink in again because it's going to get asked on the last tests. These salivary amylase also can uh, uh, chemically digest starches. The duct out of the salivary gland, the parotid gland in particular here. Here's the duct out. It enters into the vestibule of the mouth. The vestibule of your mouth is where you put your toothbrush when you're polishing the front of your teeth. So you stick that toothbrush in your mouth, in the front, in the sides, in the back, but not on the inside, on the outside of the teeth. This duct is called Stenson's duct. It's also known as the parotid duct. This salivary gland and all salivary glands are exocrine glands. That means they have ducts. And in having ducts, they deliver their secretion to a specific location, as opposed to endocrine glands, which deliver their secretion or hormone into the blood system, and it goes everywhere. The saliva goes to specific places. The salivary gland is an accessory organ to the digestive system. We're following something that might be being swallowed now, and anything that is swallowed enters into the pharynx. It's a tube that consists of three parts. Above what you can see looking in a mirror, behind the nasal cavity is the nasopharynx. The part that hangs down we see it in cartoons and such, is the uvula. That's not a part of the pharynx. It's just a marker here. What you can see looking into the mouth to the very back is the oropharynx. And when anything that is swallowed goes out of sight below the oropharynx, it enters into the laryngopharynx. At the laryngopharynx, there's a choice of which way something will go. If it's air, it'll go into the open passageway into the trachea. If it's a bolus of water, liquid, or food, the epiglottis, which isn't labeled in this picture, it's right here, the epiglottis will close and the bolus will continue down the esophagus. This is the area here in the trachea and in the nasopharynx and in the nasal cavity, which qualifies as also part of the respiratory system and will have as a lining an epithelial layer of pseudostratified ciliated columnar epithelium. The pseudostratified epithelium is associated with the respiratory system. Okay, we've gotten down here just as far as the trachea and the larynx, but portions of the bronchioles and the larger bronchi have the pseudostratified ciliated columnar epithelium as well. A part of the GI tract that things enter into next is the esophagus. The movement downward in the esophagus is done by sequential contraction of the smooth muscle wall. And this motion downward towards the stomach is called peristalsis. I'm hoping that a lot of this really sounds familiar and I'm not going too fast. If I am, you can butt in and tell me. The upper half of the esophagus, that's the portion showing here, is lined by the stratified squamous epithelium. That's the same type of epithelial type that we have on the epidermis. Okay. Stratified squamous epithelium. It's also under voluntary control, so we can spit something back up if it's too hot, tastes too acidic, or something else alerts us that we shouldn't be swallowing this. Once it gets past this first half, it's on its way to the stomach. And then the distal half, which is the lower half of the esophagus, is lined by simple columnar epithelium. The entire rest of the GI tract is lined by simple columnar epithelium. If you're printing these pages out for yourself sometime, that's a statement that you really should underline. The colon, the small intestine, the stomach, okay, they're all lined by simple columnar epithelium. Here's a cat, heads up here at the pointer, and here's the trachea. The trachea is anterior, or in the cat, ventral to the esophagus. The esophagus lies behind it. With this probe, the esophagus has been lifted up to show what it looks like. It's not very big when you are looking at it in a picture like this, but it can expand considerably. Here's the stomach. The principal function of the stomach, which is reached next, is storage of food. It's also responsible for mechanical and chemical digestion. Chemical digestion, pretty well limited to digestion of proteins. Mechanical digestion, not limited to anything. Anything can be mechanically digested or broken down in the stomach. The stomach is lined by simple columnar epithelium. I said it, but I didn't need to, because in the previous paragraph it says everything below the first half of the esophagus is lined by simple columnar epithelium. Coming into the stomach from the esophagus, there's a sphincter muscle, a muscle that closes like your eye or your mouth if it's puckering. Okay. The cardiac sphincter, it's there to allow material to come in, but hopefully to keep it from going back out. When it does go back out, we call that antiperistalsis, if we have the time to say such a long word. But when you're in a hospital setting, you're going to call it vomiting, no matter what. I always try to avoid that word myself. Bad experiences as a child, having to clean up after another kid at school. One day I was a flag monitor, another week I was the window monitor. I had to close the windows if we had an air raid because somebody was going to bomb from the aircraft above. We had bomb drills, and the windows actually had to be opened uh, so that the glass didn't blow in every direction. Well, we never saw that happen. And then one week I was the vomit monitor, and that meant I had to go and clean up after every student that got sick. Fortunately, it was over within a week, and you only had to do it once in the semester. Teacher wouldn't do it. Hell no. Okay, inside of the stomach, here's the rugae. Parts of the stomach, this is the fundus here. Okay, this is the uppermost part. It often extends higher than the cardiac sphincter. Then the cardia, and then the pylorus. I see these as making an L shape, the cardia and the pylorus. And we call the two of them together the body of the stomach. Some people really don't have much of a fundus. It is on the test from my recollection. Others have an exaggerated fundus. If you drink a Coke or something carbonated fast and end up burping, well, why did it just come up? It was accumulating in the fundus. The exit from the stomach is through the pyloric sphincter, another muscle that being a sphincter muscle, closes circularly. The pyloric sphincter is the smallest aperture, the smallest hole, the smallest passageway that food or digesting food has to pass through in the GI tract. Okay. So something's got to be beaten down pretty well already to get through here. This is the greater curvature of the stomach. I think it's going to be labeled in a bit. And here is the lesser curvature of the stomach. It's on the medial side. On the lateral side is the greater curvature. After leaving the stomach, the digesting food enters into the small intestine. Okay. Now, where do we do that? Right here. This is the first part of the small intestine, the duodenum. The stomach is out of play here. Food enters into the duodenum and curves around underneath the pancreas. And that's the full length of the duodenum. To help move the food forward are these ribs or folds in the wall of the small intestine. In the stomach, they were larger folds. They were called fundus. Here, they're called plicae circularis. Plicae circularis. That means circular folds, and some books will call it circular folds, but on our exam, we will use plicae circularis. Well, we've got some other things here. Got another accessory organ. This is the liver. They're not showing much of the liver. They've got a piece from here down to here. This is the liver. This is the gallbladder. You can barely see the small duct out of the gallbladder, known as the cystic duct. I have another picture for you. The hepatic duct, unlabeled, comes from the liver. Where the hepatic and cystic join, you have the common bile duct. But we have a better model for that. This was just to be complete. Spleen is shown over here, and pancreas here. Pancreas is a very thin organ. It's almost like tissue paper thin, but stronger. You're not going to tear it apart just running or jogging or something like that. But it's so thin, it's almost impossible to operate on it.
Is the pancreas's only job secreting hormones?
No. No, the pancreas has a digestive function too. It secretes pancreatic amylase. It'll do it through these ducts. Okay. Remember, anything that secretes hormones is secreting from a ductless gland, an endocrine gland. Since there is ductwork here, a lot of it, in fact, we'll be talking about it in just a moment. This is an exocrine gland as well. The pancreas is both part of the endocrine system and part of the digestive system. Okay. Most of the carbohydrate that is digested in the small intestine, and only because of the secretion of the pancreatic amylase from the pancreas. Okay. I think if this picture were thrown at you on the test and we hadn't talked about it at first, it wouldn't be fair. Okay. This large object is the stomach. Here you can see ribs. It may take, on your own, the very first time without a real cat in front of you. It may take quite a while to figure out what's going on. This is liver. Here's the stomach, and here's the exit right here. That's where the pyloric sphincter is, and then the food moves into the duodenum. If I were looking at this picture, I would wonder, is this a diseased cat? Has it got an infection or something here? No, that's sometimes the actual color. Here are the rest of the small intestines over here. The first part of the small intestines is the duodenum, then the jejunum, and finally the ileum. Now, this, as we saw the model of the small intestine in the classroom, this was written in that first semester when we were back in the classroom and then had to leave. So this got caught right in the middle. It's a meaningless sentence. Okay. The villus, like the brush or a filament of a hairbrush, sticks into the small intestine lumen. Okay. Here are villi, villi, villi. Now, if you picture in your mind the circular lumen that we drew, I also drew villi and put on the outside of it microvilli. The microvilli is an epithelial layer. Since this is a part of the GI tract and not the upper esophagus, this epithelial layer must also be simple columnar epithelium. We pay hundreds to thousands of dollars for some of the models that we have. I don't like this one, but it's the only one we have of a villus. And some of these cells do not look simple columnar. These actually do look simple columnar, but some of them look simple cuboidal over here. At this point, you have to use your intellect, your memory, and just come up with the law, if you want to call it that the GI tract is lined by simple columnar epithelium, except for the upper esophagus. Inside the middle of the villus is the lamina propria. These are the capillaries that absorb nutrients from the lumen of the small intestine. And in each villus, there's also a lacteal. It will be at a different level, so I can't show them at the same time, and the model doesn't either. Okay. Here's the lamina propria, and behind it, let's say, is a lacteal. Lacteal always shows up on examinations. The lacteal is there as a vessel to absorb lipids. Lipids have to be broken down first, emulsified, and then the lacteal can take them up. Just sort of a hint here is that we can always identify the duodenum because it connects to the stomach. That's assuming, of course, I show you the connection. I would. Okay. The third section always ends at the ileocecal valve, which we'll see in just a moment. The distinction with the jejunum is not as clear, okay, and will not be asked. Okay, let's cut this down a little bit. Okay, here's the colon, and at this point here where my pointer is the ileocecal valve. The part of the small intestine attached to the ileocecal valve is the ileum. The ileocecal valve is also a sphincter muscle. Once food moves into the colon or large intestine, there is no further digestion. Okay. The main site of digestion was the small intestine. Consider the first part of the colon is this sac down at the bottom here as we stand up. This is the cecum, and coming off of it is the appendix. Some people think the appendix is a vestigial organ. In other words, it's evolving out. We don't need it anymore. We may have needed it in the past. Most people don't have any use for it. Some books are coming out calling it a possible endocrine gland. Any structure you can name, there's a book and an author that thinks it's an endocrine gland. Well, the whole body interconnects with itself chemically. And so they could be right, but it's not surprising. If I were to actually get an honest answer from you guys as to how many people have had their appendix out, I probably would find that there's about 3 in the classroom. And if I count myself, it's 4. I had mine out when I was 8 years old. And I told you this story I know about our 4-0 design teacher who waited too long. He had pain on the lower right side. You get over, and especially accompanied by any fever, you know, get to a hospital if you can. Okay. Ileocecal valve enters here into the first or second part of the, I'm not going to ask you what's the first part of the colon, because some people will say cecum and some people will say ascending colon, this part. I'm not going to get into that. The ascending colon goes up and then curves sharply. Where it curves here is the hepatic flexure. It's not written down here. I have it written down elsewhere. And then we have the transverse colon, the bend at the spleen, the splenic flexure, and then the descending colon to about here. And then it dips into the pelvic brim, into the pelvis itself. And at that point, it's called the sigmoid colon down here. The bubbly areas seemed all lined up here, containing material, and those are called haustra. Formation of haustra causes the movement of material to be sped up. The function of the large intestine is the reabsorption of water and the compaction of waste. I don't mind saying it again. That's in the nature of a review sometimes. This white line or gray line or whatever color it is meant to be a band of smooth muscle called the teniae coli. There are 3 such bands. You're only seeing one. Where's the other 2? One of them is right here, attaching the haustra to the small intestine, and one is back here. The 3 bands do not contract at the same time. Because they don't contract at the same time, they cause puckering of the colon itself and the formation of these haustra. I've seen videos of the large intestine. You can find them on YouTube too. The haustra do not look as uniform as they do in this photograph, but pretty close. Now we get back to a real specimen, and all of a sudden, all that prettiness of the model disappears from us. Okay. Well, what can we tell? How do we break this down? First off, I think all of us would probably assume that this is small intestine. it's come out or been brought out. Okay. If this is the small intestine, follow it around, and we see one end going in right here, and this is the ileum going into this structure, which is the first part of the small intestine, large intestine, and that's the cecum. This is the large intestines going up and away, be coming down. This is the ileocecal valve's location. You cannot see the valve without cutting it, but the location is where these two come together. The liver, okay, located under the diaphragm. The head of the cat is over here on the right. Here are lungs. Here's the diaphragm, our prime mover of respiration. And here's the liver right here. Sorry, right in here. Here is the liver. This guy here that had the needle on first, this is the spleen. This little guy is the gallbladder. There are several functions of the liver that we mentioned previously. One of the functions of the liver in the digestive system is to produce bile salts. Bile salts help emulsify fat. Fat is not digestible, but it is emulsifiable. It can be broken down to smaller and smaller parts until they're so small they can be absorbed by the lacteal that we mentioned a moment ago. We need bile for that to happen. The bile is produced by the liver and exits to a small storage area in the small intestine. You'll see it next. And when that storage area fills up because there was no need for the bile, you hadn't eaten anything like mayonnaise or butter or anything fatty. If you hadn't, the bile won't be released. So the bile backs up and it backs into the gallbladder, which stores excess. It'll swell up considerably. If you do swallow a sandwich that has butter or mayonnaise on it, once that material passes by the storage area down, which I'll show you in a moment, passes the storage area in the small intestine, it will be released. It'll be released in mass to digest or emulsify, the proper word, the lipid. Okay. This model throws everybody the first time they see it. It's like, what am I supposed to do with this? So you look around for a chart that has a picture of it and has everything named, and you don't find it. Okay. The first time I saw this model, it was many, many years ago, but I walked into class and they had this new model, and I was wondering, what the hell am I going to do with this? I don't even understand it. Well, it's not a hard model at all. It's a model of the liver, and you're standing looking down on the liver that's been cut, and you're standing in front of the liver here. Where this seal is would be where you would be standing. And even that doesn't matter. First off, the liver's first function isn't really the production of bile salts. It's an important one that we just mentioned. The first thing the liver does is to detoxify the materials that the small intestine has absorbed. The small intestine absorbs nutrients, some of which may be poisonous. I'm not talking about somebody intentionally taking poison or being poisoned. I'm talking about just things in nature, like potatoes, which have poison in them. They have solanine in them. A green potato can make you sick if you eat a potato that's skin of which is green. But if you eat a regular potato, you still get solanine, but the liver can take care of that. The absorbed uh, material, the absorbed digested material is taken up by the small intestine and transferred to the hepatic portal vein, not into the general circulation, but instead to the hepatic portal vein, the vein that is transporting the digested, absorbed food to the liver. Think of it again. Hepatic is the liver, portal transporting vein. Okay, so all the stuff that enters into the blood of the hepatic portal vein makes its way to the liver and comes right in here. And now it spreads out. We got a lot of liver to work with. We can't just have one little lobule of the liver functioning. So it's spreading out here. Tell me the color, please, right here.
Great.
Gray. Okay, thank you. And so it moves through all these gray vessels. That's to deliver it throughout the liver. This is blood that's come in. It's probably good, but it probably has a few things that we don't want. And then the liver lobules detoxify it. And once they do, it passes through a set of capillaries, as in any circulatory system, and enters into the veins. These are the veins. This is detoxified blood. The bad stuff, if there was any, is taken out and transferred to these veins. These are the hepatic veins. We had the hepatic portal vein, and now just the regular hepatic vein. And that takes the blood detoxified into the vena cava. Here's the vena cava, which runs along your back, and not your front. So that's why this is the back, and this is where you're standing looking at it. The liver, as an organ, has to have a blood supply of its own. It has to have its share of the blood that's moving through the body and is already detoxified. The liver is not going to live on toxic blood. So here comes the blood supply into the liver right next to the hepatic portal vein. And this is the hepatic artery. The hepatic artery, you can see it deep red, spreading out throughout the liver, nourishing the liver, gas exchange taking place. And then once that has happened, capillaries again are crossed, and the blood that was always good, not being detoxified, now enters into the same venules and veins and goes back into the vena cava. There's two ways to bring blood to the liver: the hepatic portal vein and the hepatic artery. There's only one way to get it out, finally, and that's the inferior vena cava. The gallbladder shows nice in this model. And this bit of tissue is one of our serous membranes, the falciform ligament. You can't see much of it. You don't see its full length going all the way around the liver in this cut section, but that's what this is. The lobule is the physiological unit of the liver. This is a lobule right here. It always reminds me of a grain silo. But there'll be, in this case, 1, 2, 3, 4, 5, 6, 7 grain silos, most of them cut open for us to see inside. The lobule, though, is the main unit of the liver. When you cut it, like this one over here, now let's use this one here. You notice there's a vein in the middle. It's called the central vein. This one's quartered and enlarged here, the central vein. Blood is brought to all of the lobules, and it enters into the cells at the periphery of the lobule, and the blood heads for the central vein. As it's on its travel to the central vein, it is detoxified by the cells of the liver, and the next diagram will show you that better. Okay. Here's that central vein, cut in quarters. Okay. At this point here, we have 3 structures. Here's the hepatic artery. Here's the hepatic portal vein, simply called portal vein here. Now this one's coming from the small intestine. This one's coming from the arterial circulation of the normal circulatory system. Hepatic artery, portal vein. And also we have a third structure, a bile duct. That's bringing bile from the liver itself and taking it away towards the gallbladder or the hepatic duct, eventually to emulsify fat. The bile that's produced in the liver has to get out of the liver, and it gets out through the bile duct. Now, if I back up for just a second, 1, 2, 3 lobules, and something in the middle here. 1, 2, 3 lobules, and something in the middle here. What's in the middle is a triad, and this is the triad. 1, 2, 3. Portal vein, hepatic artery, bile duct. Around any lobule, which is 6-sided normally, this one looks very nicely 6-sided, there'll be 6 triads. 1, 2, 3, 4, 5. The 6th one would be here. Liver, largest gland of the body, and all glands except the neurohypophysis are composed of a tissue type called glandular epithelium. So that makes it easy as far as the histology portion is concerned. You're just going to learn it as glandular epithelium, unless it's the neurohypophysis, in which case that's nervous tissue. Here's a summary I threw together, listing the things that we just looked at and the type of material, the type of tissue they're composed of. Parotid glands, glandular epithelium. Upper esophagus, stratified squamous. Lower esophagus, stomach, small intestine, and large intestine, all simple columnar. And the liver and pancreas, both glandular epithelium. Okay, any questions? The recording for that's going to be, of course, posted this evening, or as soon as they get it to me. They've been getting it to me very fast recently. I don't know why, but so I'll get that posted to you this evening. I'm going to give you a break for right now. When we come back, I'm going to start the review of the nervous system, and probably do 2 of the 3 parts because they're the easiest and go together the best. Save the other part for Thursday, and then be able to cut off and allow you to take that quiz, the endocrine system quiz. Some people may not know about the quiz because some people weren't here on last Thursday. And I hoped that if that was the case, they would read the recording over the weekend. But it doesn't look like the recording was used but twice over the weekend. And so you want to keep yourself abreast of things. You may need to get yourself a partner if you think that there's any announcements you need. Okay, I didn't. I didn't send out an announcement for a bonus quiz. I've sent out announcements for everything else, and I will continue. Okay, but if you're up on the material, you'll do fine. Bonus quiz is something that's not needed to score an A in the class. It's nice to have the extra points in there, but you don't need bonus points. So, okay. Unless you have a question, let's take a break until 3:30. Okay, it's 3:17 now, 3:30.
Thanks, Charlie.
Okay, be right back. Okay, finally back. Sorry, lost track of time. Anybody there?
Yes.
Yeah.
Hello.
Okay. Okay, reactions. Okay, see if we're still connected. Oh, I have to go through this again. Can you see my cursor?
I only
No.
see you, Charlie.
No?
I don't see anything but you.
Okay. Okay, now we're back at week 14.
Yes.
Yes.
Yeah. Yes or no? I heard both.
Yes.
Yes.
Okay. I've got to find out. There's got to be a shortcut to getting back on. I mean, I have to go out and log in again every time. Well, we'll make do. Okay. What I was going to do is start the sort of a review of the nervous system, not the spinal nerves, but back 2 weeks. Autonomic nervous system, nervous tissue. Okay. You said that as a group that you are having more difficulty with the nervous tissue. Well, what I'm going to review today isn't the stuff that is giving you the trouble. I mean, I don't think it is anyway. I think it's the spinal nerves. But we can't just jump to the spinal nerves and disconnect them from everything else. So we're going to start at the beginning. We've got plenty of time to do this. I don't have a lot of lectures left to hit you with or anything like that. Okay, so I'm just looking for myself, for my benefit. I'm scrolling around. Okay, this wasn't on 10/20, obviously. This was just the old date. Okay, functions of the nervous system: conducting stimuli towards the central nervous system. Okay, that's one thing. Conduct motor impulses away from the central nervous system and to maintain homeostasis. When we say conduct sensory stimuli, that means sensations, obviously. Uh, Sensory stimuli are also afferent stimuli. Motor or movement impulses are going to be going to the muscles. And these are efferent stimuli. Afferent and efferent can be added in here. Okay, so we have to have input. We need some input from the environment. The afferent nervous system brings that input in. The central nervous system must be reached before any reaction can take place, whether it's a reflex arc or if it's a thoughtful response. And then we need to, if we want to act upon the impulse coming in, we need to send a motor impulse out. If we can't do that, then we can't react to it. We're not essentially paralyzed. The components of the nervous system include the central nervous system, which you know is the brain and the spinal cord. Okay, everything must reach the brain or spinal cord to get a reaction of any sort. The peripheral nervous system is everything else. That includes the afferent nervous system, the incoming nervous system, in other words, and the efferent nervous system going outward, which is motor neurons, motor nerves, the autonomic nervous system, and its two subdivisions, sympathetic and parasympathetic, and the cranial nerves. Here's the chart that shows that. The brain and spinal cord are up here at the top and comprise the central nervous system. Afferent fibers or sensory neurons bring information in towards the CNS. Efferent fibers or motor neurons carry information away from the CNS. The motor neurons, however, are divided here. Some go to the somatic nervous system. That's the voluntary nervous system, the one you and I have control of. That'll be primarily skeletal muscle. You may think you have control, and I do, of some cardiac muscle, for instance, and smooth muscle, but it's not allowed here by this chart or any book or any examination. So the efferent fibers, the motor fibers are divided in two. And one of the divisions is the somatic nervous system, also known as the voluntary nervous system. The other is the involuntary nervous system, and that's also known as the autonomic nervous system. The autonomic nervous system, we have since learned, is divided into two parts as well. The sympathetic, are also known as the thoracolumbar system, and the parasympathetic, the craniosacral. Okay. Although the spinal cord obviously runs through the neck and the 7 cervical vertebrae, okay, the 7 cervical vertebrae, they have nothing to do with the sympathetic or parasympathetic nervous system. Okay. If they did, their name should be in here, but it's thorax and lumbar, cranial or head, and sacral. You don't see the word cervical in here. Okay. Glial cells account for about 90% of the cells in the CNS. These are the supportive cells. The astrocytes are one of those types of cells. Astrocytes are necessary. They're like the glue in the brain. You can't just have a loose bag of neurons. You can't just have a loose bag of or box of spaghetti. These strands, these neurons are all held together, held in place. Here the astrocyte is in, I'm going to call it a gold color, okay? And it's attaching to neurons and also to vessels. Okay, this is a capillary. It's the cement in the brain, the astrocyte. The oligodendrocytes, these small guys here, in this diagram, they're surrounding a multipolar neuron, one that has a lot of entrances, dendrites, and a lot of X's, axon branches, axon terminals, also known as teleodendria. Teleodendria. These oligodendrocytes, uh, just let me get away with the color differences. It's blue to me. Have extensions that wrap around the axon branch of the multipolar neuron. Okay? Now, this area here would not be vacant. This area here would also have another oligodendrocyte doing the same thing. I guess they have removed that to show you the axon, or the axon cylinder, as it's also called. The oligodendrocytes is sheathing, forming a myelin sheath around the axon, causes the impulse that moves through the axon to speed up, to speed up. You ever hear the expression, hurry up and wait? You know, you race to get to someplace, and then you get there and there's a long line. Hurry up and wait, hurry up and wait. Doesn't do you
The
any
big thing in the military.
good. Yeah. Uh, okay, so this is hurry up. The myelin sheath causes to hurry up, but you get to the end of the teleodendria, you got a synapse, and you got to wait. Now, in your terms and my terms, it's a fraction of a second, milliseconds. But in the scheme of the speed of the nervous system, it really is a slowdown point. Slowdown point. Okay. Well, we move them faster here because of the oligodendrocytes. Microglia, microglial cells. These are the cleanup cells. These roam the brain and the spinal cord. They can be attached and in place permanently, but many of them move around looking for bacteria, anything that shouldn't be there, a burst capillary that has coagulated but left uh, uh, red blood cells behind, dead red blood cells. The microglia is here, astrocyte here. This is the oligodendrocyte. And here was the example of the neuron, which happens to be multipolar. Multipolar neurons are the most common type. We're going to do that one first. Types of neurons. Okay. Neurons that travel together doing the same thing, redundant, in other words, many of them, are said to be a nerve. It can be an afferent nerve that's incoming towards the CNS, efferent nerve, outgoing or motor, and it can be a nerve that runs in both directions, in which case it's a mixed nerve. Most nerves in the body are mixed nerves. Okay, here's a textual description of the first type of neuron, the multipolar neuron. Okay, I'll leave that for you to read. I did last time too, because I'm going to say everything that's in there. Here's the multipolar neuron down here. The body is also known as the parakaryon. Parakaryon. Sorry for moving it around on you. Don't worry about the mitochondria inside of it. These incoming structures that look like coral, these are dendrites. Each one of them can bring an impulse inward towards the parakaryon. No one of them would be sufficient to excite the parakaryon and its axon hillock into propagating an impulse. You need the cumulative depolarization from several dendrites to depolarize this portion here called the axon hillock. When that happens, when you get enough, then you've got an impulse that'll move along the axon cylinder and move faster if, in this case, it has a myelin sheath. The node of Van Veer are just the slight spaces between the portions of the sheath. Now the term Schwann cell. The oligodendrocyte wraps around the axon as it did in the previous diagram. The Schwann cell is also capable of wrapping around and forming a myelin sheath. And in this diagram, they purposely put a Schwann cell in so that you realize both of them do that. I don't think it's quite as obvious as they thought, but the Schwann cells wrap around neurons, wrap around the axons of neurons that are located in the PNS, the peripheral nervous system. Only in the CNS do we find oligodendrocytes. There are no oligodendrocytes in the PNS. So taking over their duty of forming a myelin sheath is a type of cell called a Schwann cell. Then in the multipolar neuron, the impulse can be propagated through several branches. These variously are called axon terminals, or here's the word I typed it in, teleodendria, or teleodendrion, singular. In a bipolar neuron, it's a little bit different. You've only got one dendrite. Then it goes into the cell body, and it must immediately initiate an impulse down the axon to the axon branches. You do not have to have an accumulation of input to get the propagation of an impulse. You only need to stimulate the dendrite, the single dendrite, once, and it will fire. We have that situation, for instance, with light. You get one blast of light, and it fires right away. You've all been to the optometrist, and they flash a light at you, snap a light at you. If not, you can imagine it, it goes right away. Have you ever looked or stared at something long enough, though, that it either changed its color or disappeared completely? You've been zapping the same dendrite over and over and over and over and over, and it needs more energy, and it doesn't have it yet. So it quits working temporarily. The term pseudo-unipolar neuron is actually fairly recent. It used to be just called a unipolar neuron, and all was well, but somebody wanted to be more perfect, and that's fine too. And so we have to relearn it. You don't have to relearn it if you never had it before. I had to relearn it, so pseudo-unipolar neuron. Okay, and now that's incorrectly called a unipolar neuron. Let's look at the picture. Two extensions. There's a Schwann cell involved, so we know that they drew this picture diagram or even cartoon in the peripheral nervous system, because you only have Schwann cells in the peripheral nervous system. You only have oligodendrocytes in the central nervous system. Here's the cell body. Okay. Here are the dendrites over here. They're all coming together as one first, though, not at any axon hillock. They're all coming together, and they travel for some length, myelinated by a Schwann cell or a group of Schwann cells. Come into the cell body, which is the perikaryon. Do not worry about the word Nissl body. Okay. And then out and continuing on through the axon, here's the myelin sheath here. Now, if you look at this picture, myelin sheath points to that, Schwann cell points to that, and it sure looks like the same thing. It is. There's one way in and one way out, but the cell body is not dead in the middle as it was in the bipolar. One way in, one way out cell body in the middle in the bipolar. Where do we find these pseudo-unipolar neurons? Primarily, they're in the afferent nervous system, coming from the body and body surface and going into the central nervous system. They're touch receptors, they're heat receptors, they're pressure receptors. Okay.
Is there a specific reason why they're found in the afferent system as opposed to elsewhere?
No, you're going to have to ask God that one. That's the thing with anatomy. The first thing you do in anatomy is you catalog everything you see and where it is, and then the physiologist comes along. Maybe it's the same person, and tries to figure out what it does. Okay, there doesn't have to be a rhyme or reason or a connection between the two. I don't know of any reason, myself anyway, why these are only found in the afferent nervous system and they aren't found elsewhere. Okay? I don't know of a reason why they couldn't be found elsewhere. The fact that they're not is just a fact. Okay, that's all I can tell you. Sorry.
No worries. I figured I'm pretty sure my future professors are gonna really like all of the questions that I have. All of them are basically physiology questions.
Oh God. Okay. Be sure you know what a nerve is. Bundle of axon fibers. Bunch of neurons running in the same direction or in a mixed direction. Nerve fiber, only a slight difference, only a slightly different, doesn't have the connective tissue around it. Afferent fibers run towards the CNS, afferent away from the CNS. Somatic means of the body, whereas visceral means of the organs. Neuron, axon, dendrite, perikaryon, we just went over all these. Now, I didn't use the word peripheral process. Peripheral process is often used in bipolar neurons here. This is a bipolar. And this is the peripheral process. Well, actually, the diagram showed it as the dendrite. And that's what it's functioning as, the dendrite. This is the axon and the axon branches. Central process is the name given to the portion leading out. the peripheral process and central process together actually function as an axon. I'm going to jump to this word, effector. I did before. It's something that is effected. For instance, skeletal muscle, cardiac muscle, glandular epithelium. Okay. Adipose is considered these days by some people to be an effector as well. But we're not including it here. Your book doesn't include it. It's just not accepted yet. I think probably it will be. Where one neuron has an axon going to axon branches and then reaches a dendrite, which is not drawn here, you have a synapse. Synapse here, synapse here. Gray matter is part of the CNS that is not myelinated. The part that is myelinated is white matter. The nucleus is not only the nucleus of a cell, but it's a collection of neural cell bodies within the CNS. So there are places in the brain where we have various nuclei that seem to be responsible for certain actions of the body. And a ganglion, a term we've used a lot, is a collection of neural cell bodies, but it's outside the CNS. In other words, it's within the PNS. Okay, there are no ganglia in the central nervous system. Okay, don't leave yet. Okay, here's the endocrine system bonus quiz. There are 10 questions. It's only worth 5 points. So you got to get 2 right to get 1 point. I always tend to round up. So if you get 1, it's, if you only get 1, you still get 1. If you get 2 right, you get 1. You get 3 right, you get 2. You get 4 right, you get 2. In other words, divide your number by 2, and if it's an odd number, round up 1. That's what I'll be doing, and I'll be sending you your score. But I want you to take the quiz and realize there's only 5 minutes. Now, the last time I had somebody concerned about a 3-minute bonus quiz. Well, I only upped this thing to 5 minutes here. If you know it, you know it. If you don't, you don't. It's bonus, and that's that. It's not like one of the other quizzes where you get 80 minutes or anything. Oh, I misspelled endocrine. Sorry about that. So when you open up the test, okay, and I'm only going to hold it open for an hour. I want you to take this test right away. You know, if you feel you're going to go study for it or whatever, it's really not necessary, and it's really not going to help much. If you bust an hour, you're not going to get a ticket at all. And then finally, sometime this evening, or right away if you want, do the 2 practice quizzes from last week. practice quiz for nervous tissue, practice quiz on tissues. One has 15 questions, one has 10 questions. Okay. The one you're going to do right now has 10 questions. You need to do these two. Okay. I know I can easily tell who has done them. And if you do those two, then I'll add in your extra credit and let you know what you got. It won't be right away that I do your extra credit because I'm going to wait till this evening to be sure I get everybody that wants to do the practice quizzes. Okay. Any questions? No
Yeah,
questions.
so I have a couple of questions, and these, like I said, might be more physiology questions, but it's on the endocrine information. So for TSH, I was wondering what exactly it means by stimulates the thyroid for its own hormone production, because I didn't really think that the glands produce their own hormones.
I don't have that page here. I'm afraid to lose the page. I guess I could. But I didn't quite understand what you said. Let me just say what I'm going to say, and then you tell me what I've left unsaid. Thyroid stimulating hormone. Okay, this is a hormone that is produced by the anterior pituitary. Okay, it leaves, gets in the blood system, and when it reaches all parts of the body, it'll also reach the thyroid gland. And when the thyroid gland is triggered by thyroid stimulating hormone, it's triggered to do what it is supposed to do. And the thyroid is to produce calcitonin and thyroid hormone, which it won't do unless, in any amount, unless it gets the hormone TSH from the adenohypophysis. Okay, now,
Gotcha. That makes more sense.
okay, I'll have to read and see what it said. See why. Okay, what else?
I was wondering what for corticosteroids, what the regulation of mineral uptake in blood basically means.
Uh, I don't know all the steroids, but generally,
That's in the adrenal glands.
huh?
It's from the adrenal glands and the corticosteroids, and it says that the effects that happen are regulates blood volume, reducing inflammation, and regulation of mineral uptake in the blood. And I was just basically asking like what it means by mineral uptake in blood.
Mineral uptake are the micronutrients. The micronutrients are not like calcium and sodium. That's taken care of elsewhere. This is more like manganese and magnesium. Okay, the trace elements. That's all I can tell you.
Okay, no worries. And then as far as estrogen goes, I was wondering basically what feedback it sends to the pituitary and like why testosterone doesn't send information to the pituitary.
Estrogen, the information it sends to the pituitary is that pregnancy has occurred, or that implantation at least is underway, and to stop producing follicle stimulating hormone, because we do not need to stimulate another follicle at this time. So it's negative feedback it's getting. And
That is
yeah,
super helpful.
and what was the other one? Did you have something?
That was really it. I guess my only
Okay.
other question is what basically is calcium metabolism and why does it only really occur for young people specifically?
No, now you're really out of my league. Okay, you're into the physiologist area. You
That's
have
right.
to.
I figured most of these were kind of more physiology questions.
Well, that's okay. A lot of times I get asked physiology questions. Now that one I couldn't answer, but a lot of times I get asked physiology questions that I can answer. But then the person gets into physiology and says, my instructor talked about it a different way or thinks of it a different way. I'm not gonna step on toes. You know what I mean? Okay, so there was one other thing I wanted to tell you guys, but I'm sure I'll think of it for Thursday. It'll be fine. Okay, so right now I'm turning on the test. I'll check when we leave here. It's on. That test is at the bottom of week 14, and then at the bottom of week 13 are the other two practice quizzes. Okay. Please, on the practice quizzes, you weren't expecting to do those either, possibly today, but just find out what you know and what you don't know. I'm not going to look at it. I'm not going to take it apart. I'm just going to see who did it. Okay. And that should work. Okay. Just one little. Okay. Oh yeah, the couple people that have already done the practice quiz. Don't worry, you do not need to do it again. Good. Okay, then I'm going to not wait for you to exit. I'm going to end it so that you can all get started. Unless you've got a question. If you got a question, you tell me. I'll let you stay behind. But I have to leave here too to attend to the quiz. Okay, see you Thursday.
Yeah.
Thank you, Charlie.
Thank you, Charlie.
Thank you.
Bye bye. Bye bye.